Isomerization's impact on drugs and also its relevance. So before we get into that, I think it's fair to discuss the rise of the pharmaceutical industry and what it is today. We categorize the rise of the pharmaceutical industry by the synthesizing or manufacturing of the first drugs. We've, as human beings, been using natural herbs and medicines for a very long time, like mint to treat upset stomachs. We use aloe vera to treat burns and skin conditions. But the first drug that was created was actually chlorohydrate. We actually manufactured this drug in the 1830s, but it wasn't realized its chemical its medicinal potential until 1869 by Matthias Liebrecht. He was a professor in Germany who discovered its sedative effects, so it was effective while being used for surgeries. Before, we used to use ethanol, which was not very good when you're getting your arm amputated. And most of the early drugs that we have that were created was actually derivatives or spin-offs of a lot of coal-like products and actually synthetic dyes. The two main spin-offs coming from aniline and p-nitrophenol. This is p-nitrophenol. This is the benzene ring with the alcohol group, and this is the nitro group. P means para, means 1,4 orientation. This is aniline. And the drugs that come from it is, for example, anacetonilide, which is this molecule right here, and phenacetin, both being analgesics, which means that they help treat inflammation, and they also help stop pain. This being salicylic acid. Does anybody know what salicylic acid is? It's aspirin. That's the pure form of it. And we have two methods of development when it comes to drug discovery. First, we have high throughput screening, which means that we test a bunch of different chemicals to determine their metabolites, as well as their carcinogenic effects, whether or not what receptors they target, and many different things like that. And this method actually tests over 100,000 compounds daily. For example, does anybody recall Dr. Wakefield's, during his presentation, he sent over the compound for testing. They found that it was actually had some benefits with targeting opioid-like receptors without the euphoric effects that typical opiates have. That's a good example of high throughput screening. Rational drug design is actually the method that most pharmaceutical companies use to develop drugs. And what this does is it actually has a specific aim and what it's trying to achieve. So if it's trying to achieve trying to target a specific receptor, they'll design the drug to do that. Examples are oseltamivir phosphate, which is otherwise known as Tamiflu. So if anybody has the flu, they'll prescribe this drug. What it does is that they designed it to attach itself to the wall of the virus and prevent the virus from infecting new healthy cells, allowing your T cells or your immune cells to target and destroy the virus before it quickly replicates itself. Enforcement, it's difficult to pronounce it, but enfortumab is actually an HIV virus, which actually has the same mechanism as Tamiflu does. It targets the HIV virus, preventing it from attaching itself to healthy T cells used to destroy viruses and detect viruses. So this is actually very effective in HIV treatment. Zolpidem, otherwise known as Ambien, is very effective, and it was designed to actually help treat insomnia and anxiety. So what they designed this drug to do is to attach itself to our GABA receptors, open up the channels, and release chloride ions into it to help disturb synaptic receptors in our brain helping slow down our brain function and releasing more GABA, calming our nervous system down. So these are all thanks to rational drug design. And these two are very relevant in the industry and to isomers because these two methods of drug development help us determine what kind of isomers are effective in treatment of illnesses. So the first type of isomers I'm going to talk about today is structural isomers. These contain the same molecular formula, however, they differ in bonding patterns. And I'll give you a few examples. These are going to be the least impactful as far as isomers go in drug development. And the reason why is because when you have two different structures or bonding patterns, they're going to have completely two different effects, two different reactivities with other biochemical impacts. They're not going to react the same, and they're going to have different characteristics, both physical and chemical speaking, like melting points. But they do have a huge impact on biochemistry, given that a lot of neurological diseases that we have, for instance, Parkinson's, Alzheimer's, and Prions disease, all really terrible neurological diseases, are caused by our proteins in our brain folding itself and actually creating new different structural isomers, which therefore starts destroying these brains and causing deterioration in functions of motor skills. And then here are some examples. Does anybody know what is the IUPAC name of this? It's butanol, typically used in as compounding in drugs like amoxicillin, which is antibiotic. It's also used in a lot of vitamins, and the reason why it's used is because it's really effective in mixing the drugs and keeping them and preserving them. This is going to be diethyl ether, and these two have completely different metabolites. For instance, butanol is going to have actually similar metabolites to ethanol, and it actually is an alcohol, so it will induce those same similar effects, but the metabolites of an alcohol is going to be aldehyde, whereas diethyl ether is actually anesthesic and it's used during surgeries a lot of times and it will knock you out. So these two have two different metabolites. The metabolite of diethyl ether will be ethanol, actually. Ethanol, and it'll break down further to carbon dioxide. These two drugs are completely different. So does anybody know what phentermine is used for? It's used to treat a weight loss, and it's really effective in doing that. It does have stimulant-like effects, but it's not known to cause any severe feelings of euphoria or to cause any psychoactive episodes while taking phentermine. However, the structural isomer of phentermine will be the notorious methamphetamine, which is, if anybody knows, really bad drug and will cause a lot of negative effects. Geometric isomers. So we have geometric isomer is going to be the same principle, same molecular formula. However, it differs in the position of functional or R groups, or functional or substituent groups attached to a rigid structure being a double bond or an aromatic structure. There are several different ways in which we categorize these. There's cis and trans meaning across, like transatlantic. Cis meaning on the same side of, it's Latin for the same side of, and these refer to the substituents. So trans, for example, this would be labeled as a trans isomer because the two R groups attached to this double bond are across from each other rather than being cis, which would be on the same side. E and Z are a similar principle, but it's more commonly used in science as it helps determine more than just two of the same functional groups. It'll be in reference to three or more functional groups. For instance, you have bromide, chloride, and then hydrogen. You'll be able to label this E molecule. E standing for Engenin, which means opposite, Z standing for zusammen. This has a specific impact on how drugs work on us, because when you do change the positions of these groups, you can also change how it's going to bond to the different receptors in our brain. and I'll get into that in a little bit. Geometric isomers, and a lot of times, are considered diastereomers, and I'll get into that in a little bit as well when I talk about diastereomers. Examples would be, this is going to be cis clopin thicksol, and this is trans clopin thicksol. So what is the important thing about cis is going to be that it treats schizophrenia. So it sedates your nervous system so that you don't freak out when you start seeing these things. It also lowers dopamine levels in the brain, which is thought to have triggered symptoms of schizophrenia. However, when you take the trans molecule, you're not going to see any effects. It's not going to medically benefit you if you have schizophrenia. Trans platinum and cisplatinum. Does anybody know what these are chemically, or what these are used for? If anybody undergone chemotherapy or known anybody that has undergone chemotherapy, they would know that cisplatinum is actually effective in stopping the fastest growing cells in our body, which is thought to be cancerous cells, from replicating. It will literally go into the DNA and stop the DNA from replicating itself. So the trans platinum, actually, if you take that, you're most likely going to die of cancer because it's not going to do anything. And these are to help explain how geometric isomers impact pharmaceuticals. Stereoisomers is going to be actually the most impactful out of all the isomers, and what it is basically 3D orientate. It's the configuration in 3D space, and what it does is it make up over 50% of the medicines. So examples would be labetalol, a lot of different racemic mixtures, as well as enantiomers that are used. Racemic mixtures being a mixture of 50-50 mixture of enantiomers, and racemic mixtures itself makes up 25% of all the drugs. And there's different types of stereoisomers. You have levo versus dextro. This is actually determined experimentally, and it's derived from how we determine these. And the way we determine is actually, here I have a light. When you shine the light, it's going to emit different rays from all different angles. You shine it through a polarized lens. You have the molecule in this little tube, whatever type of isomer it is, and what it'll do is rotate the light towards a certain direction. So if it rotates the light to the left, it's going to be considered levo. If it rotates the light to the right, it's going to be dextro. SNR is actually the best technique in which how you determine what is the specific isomer, and this is going to be in reference to the orientation or the order in which priority groups are on the chiral center. Large L or capital L versus D is actually just used for types of amino acids and monosaccharides, which are like sugars. And it's typically describing these in a Fisher projection, which I'll show you in just a second what a Fisher projection is. This is a Fisher projection, and how you determine whether it's capital D or capital L is actually the last alcohol group is actually going to be pointed, if it's pointed to the left on a Fisher projection, it's going to be considered L. If it's pointed to the right, then it's going to be considered D-glucose. This is an example of racemic mixture, and it's between levo and dextrocitrazine. Citrazine is actually effective as an antihistamine. So if you have an allergic reaction or if you are suffering from allergies, you typically are going to take a medication that contains citrazine. You have R-thiolomidine versus S-thiodomide. Does anybody know what that is actually notorious for? In the 1950s, Germans actually gave a racemic mixture of these two medications to treat morning sickness. What they didn't know is that when you take both isomers, it's going to create horrible effects. Your children are going to be deformed. So one of these isomers actually, they still use this today, but they'll only use a single isomer to treat morning sickness, whereas the other one restricts oxygen flow to the fetus, which will start causing abnormalities within the fetus. Enantiomers are mirror images of each other, and they contain two or one or more chiral centers. They're non-superimposable, and they're going to have identical chemical and physical properties. But they do differ biologically, and the reason being is that it's like a shoe. When you put your right shoe on your left foot, it's going to feel weird. It's the same thing when you talk about binding with different receptors. They're going to vary depending on the isomer. It's not going to be able to fit into a certain receptor, which is not going to be able to be effective in its treatment. Examples of how it differs biologically is L-DOPA versus D-DOPA, which is used. L-DOPA is used to treat.
Trying to wrap up.
Okay. L-DOPA is used to treat Parkinson's disease, whereas D-DOPA will actually cause terrible side effects of lower white blood cells in the body. This is two different forms of amphetamines. This is L-amphetamine, this is the chiral centers, and it's obviously going to vary where one's popping out, one's going back. This is citalopram, which is antidepressant. The chiral center is going to be located right here. And this is levo versus dextro, thyroxine, which is used to treat thyroid disorder. Diastereomers are actually not commonly used within the pharmaceutical industry. However, what they do use this for is separating enantiomers out. What they'll do is they'll use acid-base reactions to start creating different diastereomers, which diastereomers have different chemical and physical properties, which is easy to separate out rather than enantiomers. And these are examples of diastereomers, is when you actually have two similar chiral centers. Instead of being mirror images of each other, you're going to flip one but not the other. And when you do that, you're going to create diastereomers. This is an example of D-ethros, which is a sugar. This is ephedrine, which is used to treat blood pressure, but it turns into a completely different drug, Sudafed, which is actually used to treat decongestion. Racemic mixtures is obviously a 50-50 mixture. It constitutes 25% of all the drugs. You have three different categories. One category is going to have an active isomer form, while the other one is going to be less active or completely inactive. Then you have type 2, which is going to have both isomers are equally as effective of treating a certain condition. And then you have a third category, which is one effective, and the other isomer is going to be converted to the effective isomer. And racemic mixtures can be separated by biochemical processes. For example, yeast can distinguish the different enantiomers within a certain racemic mixture, and it'll actually start separating these out for different chemists and pharmaceutical industries. And you can also separate them out using the high-powered liquid chromatography. So this is group 1. This is valsartan, which one form, which is the L-valsartan, is the effective form for treating blood pressure. It will lower your blood pressure, while the D-valsartan is less active or is inactive. This is fluoxetine, which fluoxetine is a serotonin uptake inhibitor, and both isomers are equally as effective. This is ibuprofen, where the S-ibuprofen is actually the one that relieves pain, but the R-ibuprofen is actually taken and transformed into the active isomer, and it can be used to treat inflammation as well. Any questions?
Yeah, we'll just push on through. Thank you, Lucas.